This lecture is on the T cell maturation, activation, and differentiation. In the development of T cells, there are some requirements. For example, the rearrangement of the germline, the TCR genes, and the expression of different markers in the cell membrane. These developing T cells are called thymocytes. They proliferate and differentiate to become different types of T cells. So they will become different subpopulations of mature T cells. The final maturation of most of the T cells would generally differentiate into two populations, the CD4 and the CD8 T cells. So the CD4 are actually the markers that are present in that particular type of T cells, and their function is mainly to uh, interact with MHC molecules, class 2 MHC molecules. CD8 cells, on the other hand, would interact with class 1 MHC molecules. The thymus is the source of all the T cells, most of the T cells, and it is where they become different types of T cells. So this is possible because of this process called selection, positive selection and negative selection. The first one, positive selection, allows the survival of T cells with TCRs that are capable of recognizing the self-MHC molecules, and it is responsible for the creation of a self-MHC restricted repertoire of cells. The negative selection process eliminates the T cells that are too strongly reacting with self-MHC or with self-MHC plus T cell peptide. So this would this is an important factor in the generation of T cells that are self-tolerant. That means it's capable of recognizing the self. With this maturation, there are the so-called CD3 positive cells. The TCR, or the T cell receptor with an antigenic peptide in the groove on MHC molecule, is when the activation of the mature T cells happen. Okay, the problem is there is low avidity, and therefore there is a need for coreceptors and accessory membranes in order to make the bond between the T cell receptor and the MHC molecule. So this activation usually leads to the proliferation and differentiation of T cells to become different types of T cells. Okay, so in this diagram, we can see that the source of T cells actually is in the bone marrow, and then later it will migrate to the thymus. But for these naive T cells to have contact with antigens, they have to go to secondary lymphoid organs. Right, so this is an example of the, it looks like a lymph node more or less, but okay, let's just say this is a thymus. So it has the parts, the capsule, the trabeculae, the um, the blood vessels, and the lymphatic vessels. Okay, so we can see that in the medulla, okay, that is in the central part of the organ, okay, there is the presence of um, cells, but less of the cells, developing cells. Okay, in the paracortex, that is in between the cortex and the medulla, we can see the more or less mature cells, T cells, and in the cortex we can see the B cells. So they are arranged in follicular manner, in circular manner, and the center usually is the source of the germination. So we said that there are certain things that are required for the development of T cells. One of these is the rearrangement of the genes. So the possibility that the cell will become a T cell or a B cell would depend on this gene rearrangement. So more or less, they have estimated the potential diversity to be about 10 to 15 for the alpha beta TCR receptor and about 10 to the 18th for the gamma delta TCR receptor. So this would enable the cell to recognize antigens. The most distinctive property of these mature T cells is that they are able to recognize antigens only when they are attached to the self-MHC molecules. So we have said this before. There are two selection processes that happen in the thymus that allow the survival of the T cells. So the first one is called positive selection, wherein only cells that are capable of detecting the self and the MHC molecules will survive. Okay, and the rest of the cells that do not have this characteristic would die by apoptosis. So more than 98% of them would die before they leave the thymus. And what about the second one? The negative selection process will eliminate or remove the cells that are too high. Okay, they recognize the T cell, the MHC molecules and the self molecules too much. So, and then this will be eliminated, and only those cells that have capability to detect antigens in the self and the MHC. So this will result in self-tolerance. So this is the representation of that process. We can see that the thymocyte is derived from the bone marrow. It goes to the thymus, and in the thymus, it will proliferate. Okay, so it will create copies of itself, and only those cells that can recognize the MHC and the self molecule will be allowed to survive. The rest will die. Negative selection process happens in the medulla. So this is when cells that are too reactive to self-MHC and self-MHC antigen plus the antigen are eliminated. So only those cells that do not have this property will survive, and they will become CD4 and CD8 cells. So we said that this is possible it's possible for lymphocytes, T lymphocytes, to recognize the antigen only when there is the presence of an MHC molecule. The T cell receptor. So in this illustration, we can see the T cell receptor. So it's like there is something on the surface of the T cell, okay, that looks like antigen, and they have a specific purpose. It's a dimer. It's a dimer 1, 2, okay, and then there is one alpha and one beta chain. So there's a pair of these dimers, the alpha chain and the beta chain. Right, so the tail is usually attached directly to the membrane, the cell membrane, and it's made up of hydrophobic amino acids. So there is also a hinge in between, that allows the connection of the um, dimer to the tail. Both the alpha and the beta chains have the variable regions, so that means they can change, and the constant region. So the regions of the alpha and the beta chains, this region is hypervariable that can determine the specificity of the antigen. Each T cell bears a TCR of only one specific allele. Okay. One of the T cell receptor of interest are the gamma delta receptors. They are found in small population of T cells that contain delta and gamma chains instead of alpha and beta chains. They are found in the mucosal epithelia, so that means they are found usually in the respiratory and the gastrointestinal tract. Okay, and their repertoire will be dependent on the presence of bacterial and viral antigen. They have segments which we refer to as the B, the D, and the J segments. And gamma chains would have only two of those segments, the V and the J. So delta would have the V, the D, and the J, and V and J are found on the delta, gamma chains. Repertoire is limited, so the type of cell is limited. This gamma delta TCR can recognize antigen in an independent manner to MHC. Gamma delta T cells play a role in response against viral and bacterial pathogens. The TCR cell receptor, the T cell receptor TCR is associated with CD3, then it will form a complex. This accessory molecule participates in signal transduction after the interaction with the T cell and the antigen. It does not influence, however, the reaction of the antigen. CD3 is closely related to the alpha-beta heterodimer and is required for the expression of the so-called alpha-beta and delta gamma delta T cell receptor. Each of the dimer will form a complex with a CD3. We will look into that in the next slide on the cell membrane. And then the T cell receptor can be envisioned into four different dimers, alpha beta or gamma delta heterodimers. They are responsible for the antigen that will appear on the surface of the T cell. Then the CD3 dimers, which are referred to as these Greek letters, are required for membrane expression of the T cell receptor and for signal the transduction. So just take note of the role of the CD3 dimers. Class 1 MHC specific TCR together with the CD8 co-receptors generate a different signal, and class 2 MHC specific TCR together with CD4 will produce another signal. So these are the structures or relative to those cell receptors. Right, so we have the CD4 receptor, we have the CD8 receptor, gamma, epsilon. Okay, so these are the CD3 receptors below, and we have the alpha beta receptor. So they are all attached to the surface of the cell membrane. The recognition of the complex antigen MHC complex is mediated by TCR and CD3, but other membrane molecules play important accessory roles in antigen recognition and T cell activation. Some of these molecules strengthen the interaction between the T cells and antigen presenting cells. And another function is to participate in signal transduction. These CD4 and CD8 are classified as co-receptors based on their ability to recognize the peptide MHC complex and their role in signal transduction. The CD4 and CD8 cells have extracellular domains that bind with the regions of the MHC molecule on the antigen presenting cells. Let's look at that. So the smaller CD3 complex may be associated with the TCR molecule, so the dimers here. TCR is associated with the different chains of the CD3 complex. Okay, and CD3 proteins are actually invariant or never changing. They will not change, but they are involved in signaling. So when the CD3 complex are present, this is going to show up in the surface of the T cell, and it's very important for the development of T cells. They have also signals, they produce signals to the interior part of the cells that allow for interaction between the antigen and the TCR. Then we have this so-called immunologic synapse or immunological synapse. This is word synapse is, if you remember in physiology. This is the interaction between the nerve cell and the muscle cell. So there is something like a communication pathway. In this case, when we say immunological synapse, we are talking about the TC receptor, the T cell receptor, and the major histocompatibility molecules. Okay, take note that the reaction between these two is not strong. There is low avidity. Because of that, we need some molecules to help make this stronger, and those molecules are called accessory molecules, and these are the examples of those accessory molecules. CD4 combined with MHC, class 2, CD8 combined with class 1 MHC, CD2 LFA3 combination, LFA1 and ICAM1 combinations. The specificity for the antigen resides only in the TCR, and the accessory molecules are never ending, never changing molecules. That means they are not affected by the interaction. Expression is increased in the presence of the cytokines, and engagement of TCR with the antigen MHC complex is needed for the activation of the T cells. Another signal would come from the co-stimulatory molecules, which we call them co-stimulatory molecules because they participate in stimulating the T cells to be activated. So these are the CD28 on the T cells that interact with the B7-1 or the CD80 or B7-2 CD86 molecules. So again, these molecules are never changing. They are not affected in any way by the interaction. Engagement of TCR and antigen MHC complex will not actually result in any reaction from the immune system. So it's energetic. And engagement of these molecules in the absence of TCR engagement will also have no result in no response in the immune system. So these are very important. The presence of TCR and AG-MHC interaction plus the presence of the co-stimulatory molecules are both necessary for the activation of the T cells. Activation will happen only when both of these are present, and there is also downregulation if CTLA-4 interacts with B7, so that will lead to inhibition in the activation. T cell, T helper cell activation starts both as a response on the humoral and the cell-mediated immune system. So the T helper cell activation is initiated by the complex, TCR-CD3 complex, okay, wherein there is the presence of the antigenic peptide, which is bound to the class 2 MHC molecule. So these are all necessary so that the T helper cells will be activated. So this TH cell will interact with the antigen and it starts a cascade of events that makes the resting T helper cell to undergo mitosis. So they will undergo mitosis, they will divide, proliferate, okay, and differentiate into memory cells or effector cells. So the T helper cells can become memory T cells and they can also become effector cells. The resulting release of cytokines will form adhesion molecules. What are the steps that are necessary for the key T cell activation? So this is more like a summary. Number one, the antigen presenting cells must process and present the peptide to the T cells. So first, there should be an antigen present in the surface of the antigen presenting cells. And the T cells will see this. The T cells must receive a co-stimulatory signal that would come from the CD28 and B7 molecules. So there must be a signal. Number three, adhesion molecules are needed to stabilize the binding of the T cell and the APC. If you remember, we said that this binding between APC and T cell is weak. So we need some help. And this would come from the CD4 MHC class 2 or CD8 MHC class 1, depending on the type of cell. LFA-1, SICAM1, CD2, and LFA3. So those are the accessory adhesion molecules. So they help in the binding between the APC and the T cells. The signal that comes from the cell surface will then go to the nucleus and there will be production of second messengers, which are also cytokines. They will help in the cell division. It will help in the production of more T cells. This example of this would be the interleukins 1, 2, not only 2. Right, so in this illustration, we can see that this part over here would be the antigen presenting cells, the antigen presenting cells that contain the class 1 or class 2 MHC, so it depends on the kind of cell. Okay, first, this APC must process the antigen inside and show it onto the surface of the APC. And this will be recognized by the T cell, T helper cell, or T cytotoxic cell, and it will undergo activation. So when it's activated, when it will do other functions. When it's activated, on the other hand, that is also going to do other effects. Naive T cells that have not encountered any antigen or complex will just go to the circulation and the lymphatics. Until the time they have encountered the complex, they will now undergo change. So they can become effector cells or memory cells. However, not all of the cells will survive. There is what we call lymphocyte homeostasis, which is a natural way of uh, killing the excess population of lymphocytes. This process is usually by apoptosis. Okay, and what else? There is positive and negative selection, if you remember that. So that is a way also of selecting which cells are going to survive. So outside the thymus, there is also another way to kill the mature T cells, and this is through the FAS pathway. So when they are expressed together with the FAS and the FAS ligand, it will result in cell death. This process is called activation-induced cell death. So the T cell repertoire is shaped by the apoptosis in the thymus and the periphery. So the kind of cells that survive and are present in the immune system is determined by the activity of the thymus and outside the thymus. Okay, so that's the last slide for this topic. the development, activation, and the differentiation of T cells. Thank you for watching and listening. I hope you learned something.